Okay, Allison, we're good to go.
Okay. Hello, everyone, and welcome to the Invasive Species Center webinar series. My name is Allison Grant, and I'll be your moderator for today. Before we begin, I would like to acknowledge that the Invasive Species Center honors a long history of First Nations, Inuit, and Métis Nation people on the lands known as Canada and strive to show respect to their ancestors, their community, and them individually. The Invasive Species Center respectfully acknowledges that our head office is located on the traditional lands of the Anishinaabe, the Batchewana, and the Garden River First Nations, as well as a longtime settlement of the Métis people in the Robertson-Huron Treaty area. The Invasive Species Center is a not-for-profit organization that connects stakeholders, knowledge, and technology to prevent the introduction and spread of invasive species that harm Canada's environment, economy, and society. We've got a lot of great invasive species resources on our website, including invasive species profiles, best management practices, and more. So check us out at www.invasivespeciescenter.ca. You can also sign up on our homepage for our newsletter, a bi-weekly media scan and event invitations, which is where you can hear about upcoming webinars. Before we get started with today's webinar, there is a couple things I would like to mention. There will be time for questions at the end of the webinar, so if you have any questions at any time, please type them in the question box and I will read it to our presenter after the webinar. If you're having technical difficulties at any time, please type them in the chat box or respond to the email found in the registration and we'll try our best to resolve it for you. We have enabled closed captioning, so if you would like to follow along that way, you can turn on the closed captioning button on your taskbar. And lastly, there will be a brief survey following the webinar. If you could take some time to fill that out, we would really appreciate it. Today's webinar title is Hijacking Cupid's Arrow, How to Use Reproductive Biology to Manage Invasive Insects. And I'm pleased to introduce our speaker, Dr. Joséphine Queffelec. Dr. Joséphine Queffelec conducted her PhD at the Forestry and Agricultural Biotechnology Institute, FABI, at the University of Pretoria in South Africa. Her research focuses on the reproductive biology of the invasive woodwasp Sirex noctilio, and specifically how reproductive traits affect the invasion capacity of the wasp in South Africa. After completing her PhD, she conducted a research project at the FABI as a postdoctoral research fellow and started working on bark beetle chemistry ecology while continuing her research on Sirex noctilio. She is now based at the Great Lakes Forestry Center, where she conducts research on the integration between reproductive biology and the establishment capacity of biological control agents introduced to manage invasive insects. Thank you so much, Joséphine, for joining us today. And with that, I'll pass it over to you.
Thank you very much, Allison, for the warm welcome. I hope you can hear me. And thank you everyone for showing up today. I'm always excited to be talking science with people, so this is great. I really hope you have a good time. Okay, so let me share my screen. So if I don't hear anything, I'm going to assume that you can see my screen, and I'm going to put full screen. Okay, so today I will be presenting work that I did during my PhD, as I just started a postdoctoral position here at the Great Lakes Forestry Center, but I don't have much to talk about yet because I just started. So I will be presenting mostly on my PhD work, which focused on the reproductive biology of an invasive wood wasp in South Africa. So I'm French, but I did my PhD at the University of Pretoria in South Africa at FABI, the Forestry and Agricultural Biotechnology Institute. And for those of you that are interested more in invasive plants than in invasive insects, I have an interesting fact about Pretoria. So you can see on these pictures, those purple trees are Jacaranda trees. They're an absolutely beautiful view if you ever visit South Africa. October would be a great period to go see them. It's spring, so they're all in flower. And there are thousands of Jacaranda trees in Pretoria. There are so many of them actually that some people call Pretoria the Purple City. And it is truly now considered a landmark and part of the cultural identity of Pretoria. However, outside of the city, Jacaranda trees are considered as invasive. So in South Africa, they have this dual status of cultural landmark, but also invasive species that severely needs to be controlled and is actually illegal to be planted. So I did not do my PhD, as I mentioned, on Jacaranda trees, even though they're really beautiful. I was focusing on Sirex noctilio, the Sirex woodwasp. So Sirex noctilio, the females lay eggs in pine trees, and while they're doing so, they're also introducing in the wood phytotoxic mucus and a symbiotic fungus. And these two agents together can modify the physiology of the tree and provide the right growing condition for the larvae, as you can see here. And as they grow, the larvae will then dig galleries within the wood, and that would eventually lead to a decrease in wood quality. And in times of outbreak, Sirex noctilio can even lead to the death of previously healthy trees. So in the Southern Hemisphere, as you can see, in South America, South Africa, and then Oceania, Sirex noctilio is considered a major pest and just like is a cause for major economic loss for the forestry industry. So as you saw by my title, today we're going to talk about the interaction between reproductive biology and invasiveness. And there are many ways that you can look at this. You can look at it from a practical perspective, something that we're going to look at it a little bit further. But you can also look at it from a theoretical point of view, because reproductive biology, or reproductive traits rather, really influence, they can influence fitness, and so they influence also population dynamics and population diversity, so they have an impact on adaptation in a new environment. So it's also very important to know the reproductive traits exhibited by an invasive species. So to give you an example, there is this little ant that I find absolutely fascinating, called Paratrechina longicornis. So this ant is from South America, but partly because of its reproductive biology, has become now a massive pest in many countries. So what's interesting about the reproductive biology of this ant is that the queens are haploid. So it means that they only have one version of each chromosome. And these queens, to produce other queens or daughters, reproductive daughters, they produce haploid eggs that will then develop into another haploid reproductive female. Now, if they want to produce workers, they will use a haploid egg, fertilize it with a haploid sperm, and then produce a diploid worker. So that is how they differentiate between castes, is the ploidy of the individual. Now, what's very interesting about this species is that to produce males, the queens will actually produce an aneuploid egg, so meaning that there is no genetic material in it, and then fertilize it with a haploid sperm, and that will lead to the production of a haploid male that is genetically identical to his father, but does not share any genetic material with his mother. So now if you take a queen and you take her to a new environment, first of all, if she has been fertilized previously by a male and she starts a population on her own, where other species would have problems with inbreeding because of the low genetic diversity, in this species, as you can see, the genome of, or the gene pool of the females and the gene pool of the males are actually never recombining in reproductive individuals. So that means that you never have a negative effect of inbreeding on the population. So basically, one fertilized female can then start a population by herself. But what's even more interesting about the species is that it has the capacity of using the sperm of a different species. If the queen has not been fertilized and meets a male from a closely related species, she can mate with him and then use his sperm to produce workers. That is called sperm parasitism. So she will, on top of that, produce males that are not of her own species, which is incredible. But those workers now have basically the genome of two different species, they're hybrid, that have an ecological niche that is covering the ecological niche of the mother species and of the father species. So now you can imagine how that helps with adaptation in a new environment. So that's why paratrichina longicornis, or partly why it is highly invasive. So now let's talk about what I did during my PhD. So first I will present on one of the chapters of my thesis that was looking at mate choice in Sirex noctilio. So we studied mate choice because we still struggle to rear Sirex in the lab. So because there is a lot of things that we don't know about how it reproduces. So it's important to look at that type of behavior to understand how we can have better rearing systems. But also you'll see that it will have application later on for new technologies and new management strategies. So what's interesting about the behavior of Sirex noctilio when it comes to mating is that the males will form what we call leks. So there are large aggregations of males. You can only see a few males here, but sometimes you can have hundreds of males hovering over the top of a tree or at the top of a wood pile when those trees have been cut. And the females will then enter the lek and choose a few males to mate with, and then she will leave the lek and go lay her eggs. So when we look at copulation in captivity in Sirex noctilio, we found that not only the females did not mate with all the males they encountered, so meaning that there was some form of female choice, but we also found that the males did not want to mate with all the females, indicated that there might be male choice as well in Sirex noctilio. So we wanted to investigate that further. So first of all, we wanted to see if there's an effect of age on male mating success, because in Sirex noctilio, the males will emerge out of the wood anything between 7 and 15 days before the females. This means that during that time, they are basically exposed to elements and predation that can reduce their lifespan and eventually prevent them from ever mating. So we really wondered why they would leave the safe space that is the tree. And we would assume that it's probably to basically show off their fitness and show off that they can survive that long without mating. Now, in most insects, especially in species where with short lifespans, like Sirex noctilio, where the females survive between 5 days and 2 weeks, the males will usually pick younger females to mate with because they want to make sure that the females have enough time to use their sperm, basically. So we were also expecting an effect of age on female mating success. Now, on top of that, we also looked at size on all individuals, because larger individuals are usually fitter because they have, in females, they have more eggs, for example. And in males, they're also showing off their fitness. So for females, it's an indication of fitness. So larger males will usually mate more. And finally, when you observe the copulation, we often see that the larger males struggle to fertilize the small females because they struggle to bend their abdomen enough to fertilize those females. So we're also looking at an effect of size ratio between the male and the female on the mating success. So this was our experimental setup. We had 1 female and 10 males in a cage, males of different age and size, and then we recorded the number of mating attempts from each male towards the female, and then the number of mating between each male and the female. So when we talk about mating success, we have to remember that mating success depends on how attractive an individual is to the opposite sex and how receptive they are to mating. That's why here we will be talking about 2 variables: attractiveness, which is the number of mating attempts received by a female, and then receptivity, which is the proportion of copulation compared to the number of mating attempts she receives. So first, looking at the effect of age on male mating success, what we found was that older males attempted to mate more with the females, and they were also more attractive, or the females were more receptive to older males. So what we think happens is that the males might have to reach sexual maturity after emergence. They probably are leaving the wood without mature sperm. And this is accompanied by a female trait where they will prefer older males because, again, older males are showing that they can survive that long. So it's a proxy for fitness. So then regarding the effect of age on female mating success, we found that younger females were more attractive to males. And however, we did not find an effect of age on female receptivity. So what we think is happening in Sirex noctilio is that like other Hymenoptera or other wasps, the females switch from a mate location phase to a host location phase. And this is comes with a link between behavior and physiology. And it's possible that the males are actually able to sense, maybe using cuticular hydrocarbons, which females are in their mate location phase and which are in the host location phase, and that they just don't bother trying to mate with the females that are already in their host location phase. So now looking at the effect of size on male mating success, we found that larger males attempted to mate more with the females, but they were not necessarily more successful. However, regardless of their size, they tried to mate with, in general, with females that were larger than them. So we think that larger males, because they have a higher sperm availability, they become less picky than the small males. So they will attempt to mate with most females. However, as you can see on this picture, males need to bend their abdomen quite a lot to be able to fertilize the female. And like we observed originally, we found that if the male is much larger than the female, he just cannot, he's not flexible enough to fertilize her. So that is, he fails at copulation. Now, looking at the effect of size on female mating success, what we found was quite surprising because we found that small females are more attractive and they're also more receptive to males. And that is counterintuitive to it because larger females not only have more eggs and should be more attractive to males, but also because they have more eggs, they need more sperm. So they should be mating more in theory. But what we found was the opposite, that younger females are both more attractive and more receptive. So what we think is happening is that, as I mentioned earlier, larger males will try to mate with all of the females, small and large females. But because of sperm competition, because they have less sperm, the small males will then be more selective, and will try to mate only with the small females, because larger females are in higher demand. So you then have a mate choice by males to stay with the smaller females. And the fact that the small females are also more receptive, we think that it might be because of their small body size, they are less capable of rejecting males if they are not attracted to the male than compared to larger females. And so to prevent injuries, they will just mate. So that's what we call convenience polyandry, where they mate multiple times just to avoid injury. So to recap on this chapter, we found that age affects mating success in both males and females. And however, the effect of size is a bit more complicated because it depends on male size, female size, and the ratio between them. And overall, these findings have implication for management. So for example, for pheromone trapping, in Sirex noctilio, it has been a little bit complicated to find a sex pheromone that works in the field to trap individuals. And it's possible that the fact that age has clearly an effect on mating success, and so probably an effect on physiology, maybe has been hindering those findings. Also, sterile insect techniques and gene drives, things like CRISPR, are becoming very, very popular. And to be able to establish or use that type of technique on Sirex noctilio, we require very fit males, or males that would have a high mating success in the field. So we need to be able to understand what makes a male successful. So that has implication for the future of the management of Sirex noctilio. So now moving on to another chapter of my thesis, where I looked at Volbachia, which is a reproductive parasite. So Volbachia is a bacteria that lives in insect cells, mostly in the reproductive tract. And we call it a reproductive parasite because it can modify the host's reproductive biology. And depending on the strain of Volbachia that we're looking at, but also depending on the host, Volbachia will have different ways of modifying the reproductive biology. However, today we're going to talk about one specific way, and that is called Cetoplasmic Incompatibility. So this has been mostly studied in mosquitoes. This is why you're seeing little mosquitoes here. So in blue are infected individuals, so infected with Volbachia, and then in black you have individuals that are not infected. So what happens is that if an infected male mates with an infected female, together they form a fertile pair, they can produce progeny. And for an uninfected male, regardless of the infection status of the female he mates with, he can also produce viable progeny. However, an infected male mating with an uninfected female will actually not be able to produce progeny, because the Volbachia that are present in his reproductive tract actually kill the embryos produced by this pair or by this female. This is because Volbachia is transferred only to the progeny by the females. And so by killing the progeny of uninfected females, Volbachia then gives a competitive advantage to the progeny that is carrying Volbachia. So by doing so, Volbachia is then able to spread within populations of insects very quickly. And so Cetoplasmic Incompatibility is currently being studied in the context of pest management. So what you can do is that if you have a population of, let's say, mosquitoes with uninfected males and females, you can breed in the lab a large population of males that are carrying Volbachia. and then release that large population. And if, as long as those males outnumber the uninfected males in the field, they will then mate with uninfected females, leading to the death of the progeny and then to the collapse of the population. So this is why it's currently being investigated for pest management. And most of the studies have been focusing on mosquitoes, but in the future, it will be probably used for other pests and potentially maybe Sirex noctilio. So at some point in the beginning of my PhD, I was looking at the genetic basis of reproduction in Sirex noctilio, and at some point, and that involved looking at the sequence genome of Sirex. And at some point, I stumbled upon a few genes that did not look like they belonged to an insect or Sirex. They looked like they belong to a bacteria, and more specifically Volbachia. And so right away we thought, oh, this might be a good opportunity, maybe for control in the future. We need to know if Volbachia is there or not. Or at least if Sirex is compatible with a Volbachia infection. So first of all, we wanted to know if the genes that we were seeing in the sequenced genome belong to a free-living Volbachia, in Sirex, or if maybe they had been horizontally transferred. So the genomes of eukaryotes and bacteria contain mobile genetic elements, which are pieces of genome that can contain one or multiple genes that have the capacity to snip themselves out of the genome, and then after moving around, will reintroduce themselves back into the genome at a different place. These mobile genetic elements are very, very important for the evolution of bacteria, because actually bacteria have the capacity to come into physical contact and then exchange genes, a little bit like Pokemon cards. And by exchanging those genes, they are now able to acquire new abilities. So, for example, when bacteria acquire resistance against antibiotics, sometimes it is because of gene transfer. So the transfer of genes from bacteria to a eukaryote is not as common, but it has happened. And it has been described in the case of Volbachia, of genes belonging to Volbachia, ending up in a host, in an insect host. So we wanted to know if maybe the genes that we were seeing were actually not from a free-living bacteria, but rather from, that they were rather remnants of horizontal transfer. So to figure out what was happening, basically, if the genes that we were seeing belong to free-living Volbachia, what we would have been able to do was to amplify not only the genes that we were seeing, but additional genes that we knew Volbachia carries. So those genes are, for example, the 16S, which is a gene that all bacteria carry, but also things that are a bit more specific to Volbachia, like the FTSZ and the WSB. And unfortunately, after many tries and many different protocols and months in the lab of doing PCRs after PCR, we could not get any amplification. So now we're pretty sure that Sirex noctilio does not carry Volbachia, at least in the South African populations that we screened, and maybe in other populations in the world. Volbachia is present in Sirex noctilio. Now that we knew, we were 99% sure that Volbachia was not present and that the genes we were seeing were probably horizontally transferred. We wanted to find all of those genes that had been transferred, and we also wanted to make sure that they were actually Volbachia genes. and maybe find out what functions they used to serve before, so when they still were in Volbachia. So to do that, we first did a genome-wide searches using two methods. So first we used the BLAST. So this method, basically, you have the Sirex genome, and then you have Volbachia genomes. So that we use as reference. And then what we do is that we basically use a BLAST algorithm that will find alignments, or that will try to align the Sirex genome and the Volbachia genomes, and try to find sequences in the Sirex genome that look similar to the reference genome. And then the second method we used is Kraken. And with that method, basically, it uses the fact that eukaryote genomes and bacterial genomes have different signatures. So they have different nucleotide content, for example, and the genes have different structures. So Kraken is an algorithm that is able to say if a sequence belongs to a eukaryote or to a bacteria, and even more specifically to Volbachia. So these two methods together found a bunch of sequences, and then we did individual gene phylogenies to make sure that these were Volbachia genes, and they even showed us the function of those genes. So you can see here recombinase, DNA invertase. So what we found was 12 genes, or remnants of them, because you can see here the black marks on those genes, as well as the dotted lines here or there, show that there is extensive mutation on the gene. So, for example, this represents a stop codon, and these represent shifts in reading frame, and the dotted lines indicate that there is a part of the gene that is missing. So, see, you can see that this one, for example, has lost the front end and the back end of the gene, basically. So these do not work anymore. So this indicated that because there are extensive mutation of the gene sequence, these horizontal gene transfers are definitely not recent. They happened before Sirex noctilio was introduced to South Africa, because we also screened a bunch of different populations, and we found the same thing consistently across populations. So as I mentioned, those phylogenies were also able to tell us the original function of those genes. And here's what we found. So basically, most of the genes, 11 out of 12 of them, were basically mobile genetic elements. So for example, transposases, retrointrons, bacteriophage genes, all of these things are things that can move around genomes by themselves. They have the machinery encoded into their sequence. However, there was one gene, ORF8, which was not a mobile genetic element, which is a bacterial element. And we think that it might have hitchhacked with a mobile genetic element at some point. So here what we saw was that Sirex noctilio is unlikely to carry Volbachia in South Africa. But the presence of horizontally transferred genes indicate that Volbachia was present in Sirex noctilio at some point in its evolutionary history. And that means that it might still be compatible with the Volbachia infection, meaning that we could potentially use Volbachia as a management technique in the future. So I hope you remember that reproductive biology holds important information, not only about how species become invasive, but also how we can manage them in the future. So thank you very much for your attention, and I'll be happy to answer questions.
Thank you so much, Joséphine. If anyone wants to put some questions in the Q&A there. I have a question for you, Joséphine. How long did Sirex noctilio carry sorry, I'm butchering the name.
That's all right. So we are not too sure about that. As I mentioned, it definitely happened before Sirex noctilio was introduced in South Africa, but even it probably happened before that, because you can see those sequences are really, really, they have a lot of mutation there. And also we think that they did not happen at the same time. We're not too sure yet. So it's been a while, and I would be really interested to screen other populations in the world and try to find out if some of them still have Volbachia. That'd be really cool, because then we would have like a strain that we can use that is compatible with Sirex that we can use to introduce in South Africa, so we could like manage the populations in South Africa.
And how close are we to controlling Sirex noctilio with Volbachia?
So there are two things that need to be done. First of all, finding a strain that is compatible with Sirex noctilio. So that could come from either by finding another strain in another population of Sirex noctilio, but that could also come from a different insect species, either another woodwasp or another insect that lives in pine trees, for example, because that strain of Volbachia would be happy in those conditions. But also we're still working on a rearing system for Sirex in the lab, and to be able to release a large number of males that are infected with Volbachia in the field, we need to be able to rear Sirex noctilio in the lab. And so I have colleagues during my PhD, I started a little bit with that. So creating artificial diets and things like that. And now I have colleagues that have taken over that project and are doing some good work with trying to just figure that system out so they can breed in the lab, and then we can get them to adulthood, because that's also an issue. Yeah.
What is the biggest obstacle right now for rearing Sirex?
So they're quite finicky already with the mating. So the cages I used, you can see they're big, because they're big insects, eh? And on top of that, they need very specific conditions. So they mate in the morning or in the evening. So outside of office hours, I would say. And they need to be outside. They need UV light to be receptive to mating. So that's already quite cumbersome to have them to mate. And you also can't keep the individuals in a tight space together, because they get really aggressive. Other insects, you can keep males and females in like a box this big and it will be fine. But these, they need to be separated. They're pretty angry. Yeah, so that's already one problem. And then also getting them to adulthood. We don't have artificial diets that are working at the moment, or more working very well or efficiently, because Sirex has a symbiotic fungus, and the fungus is basically digesting the wood for the wasp and providing nutrients. And so right now, the only medium that works, you have to put the fungus to grow on the medium first. And that creates problem with contamination. And so that's why it's not really efficient at the moment. It requires a lot of work for only a few individuals. So, but I'm sure in the future we'll find a way to make it work.
Yeah, that sounds very difficult.
It's fascinating though.
Yeah.
You know, when something works, it's great.
For sure. Yeah, I don't have any questions in the Q&A at the moment, but if anyone has any questions, feel free to put them in there or the chat. Other than that, I'm trying to think of other questions. This is a really fascinating topic though.
Thank you.
If I don't have any more questions, I just want to say thank you, Joséphine, for presenting your presentation today. And thank you everyone for tuning in. The webinar was recorded and will be posted on our website at www.invasivespeciescenter.ca. Just a reminder to please take a couple minutes to fill out our survey, and we will really appreciate that. Thank you, everyone.
Well, thank you for the opportunity.
Yeah, thank you.